All right, so the very first part of the muscle contraction was a neuromuscular junction. The idea was that you have the neuron, most of the time these are called motor neuron. Motor neuron, again, hopefully you remember it from anatomy. It takes information from central nervous system all the way to the effector organ or the muscle. In this case, we're talking about the skeletal muscle. So your brain uh, sending the electrical impulses known as action potential, and these electrical impulses travel down the axon, and then you release acetylcholine. And these acetylcholine binds to the receptor, and then channels opens up, and voltage keeps changing. They are propagating along the muscle cells, and this electrical impulses goes down to this T tubule and then reaches the structure known as SR, which releases calcium. So this part was kind of like the first part, and there are a lot of animation that you guys can take a look at the neuromuscular junction. All right. I would like to talk a little bit about cross bridge cycle. In other words, all the events that are taking place inside of the muscle, all right, starting off with calcium release, what happens after that, all right, and how does this related to the why is it called cross bridge, and what is the cycle all about? Anyways, all right, it's basically is about muscle contracting and relaxing. That's what it's all about. Right, so we're going to put in our concentration on these steps and all the events that are taking place right here uh, after the calcium is released. So one of the things that is required for the muscle to contract is calcium. All right. So when you give the calcium to the muscle is going to contract. All right. So let's see how. All right. So if we are inside of the muscle, and the skeletal muscle, they have a particular organelle. So let's say here is inside of the muscle, and they have particular organelle known as the SR, sarcoplasmic reticulum, which contains high amount of calcium. There are infoldings on the muscle that are known as T-tubule or transverse tubule, where the action potential or is a pathway for electrical impulses to arrive on the SR. On the SR, there are channel, protein channel. These are called voltage gated channel. They open and close whenever the voltage changes. So these are calcium, voltage sensitive. So when the voltage changes and is coming, they are opening. And the very first step in this process is that the calcium is now released. All right, where is the calcium is released? Calcium is released now in the sarcoplasm. This is same thing as a cytoplasm, but basically because it's a muscle, we use sarco for muscle. So now there are a lot of calcium available. Inside of the muscle cell, there are myofibrils. The myofibrils, they look something like this. These are the myofibrils. There are a lot of these myofibrils available inside of the muscle cell. All right. So what we're looking at is one of these myofibrils. Okay, and you assume that there are calciums around. Okay. Now, give me one moment. I'll be right with you. All right, let's go ahead and take a look at in here. So now we were talking about the myofibrils, and there are a lot of calciums are available around these myofibrils. Each myofibrils is made out of protein molecules known as actin and myosin. Uh, if we go ahead and take a look at, these are the myosin fibers, and these are the actin or the thin filaments. It's in blue color. You could see clearly why they call thin filaments. Uh, and what we wanted to do is take a look at the structure and also the function of these two. Now, uh, what happens is once you locate the Z disc in anatomy, you learn two adjacent Z discs are called sarcomeres. So the sarcomere is from here all the way to here. That's one sarcomere right there. All right. And what we wanted to do is take a look at how the sarcomeres can get shortened. All right. Well, in order for it to get shortened, this myosin head has to attach to this actin. For this reason, this is called cross bridge formation, because it's kind of like a bridge between the thick filament, myosin, and the thin filament, actin. All right. But once they get connected, they need to kind of like, move. So that way they get shortened right over here. All right. So that's what we're going to get at. Previously, you probably know from anatomy or from the previous lecture that the myosin head, let me get, so the myosin, they are, look like something like that, like a golf club that the handles are twisted, and they have area at the tip that is attaching to the actin. They also have another area for attachment of the ATP. ATP can bind in here as well. Now, the myosin head can lift themselves, so pretend this is lift up now, all right? And they are up like that, okay? Now, that's one area that they could move, but this area right over here, they can actually bind to the area that is highlighted in dark color. These are the actin, all right? And these are areas that are binding site for the myosin head. Myosin heads are a lot of them. It's not just one, all right? So another myosin head can actually bind right over here as well. It could lift up and bind right here, right? Okay, so now, the another area on the myosin that can move is this part right here, the neck area, right before the head area. So a little bit later on is going to move right in this position, and this actin is going to be this way. So basically, it's going to pull the actin towards the center, all right? So one area that they could move is somewhere in the middle of the tail. This is called the tail. And another part is right below the head, or the neck area can be moved, all right? So again, you'll see it on the animation, or if you already watched it, that would be great, all right? And so on. However, they cannot just bind in here, because normally actin filament, they are blocked. You see, the binding site are blocked. These are the binding site, and the binding sites are blocked by another protein known as tropomyosin. Here is the tropomyosin that is blocking the active site. So in other words, if I have the myosin head, it cannot bind in here, all right? It cannot bind in here because it's blocked. Earlier, I used an analogy of, like, you put a piece of tape in front of your lock. Now your key cannot go in there. Now, in order to remove this on the side so that this will happen, in other words, binding can happen, you need to push this tropomyosin on the side. All right. So there is another regulatory protein, all right, that is controlling this as well. And once calcium comes in and bind to it, it can move this tropomyosin on the side. So calcium that we discussed that are coming in from SR, sarcoplasmic reticulum. Earlier, we had the channel open. That was a voltage gated channel, and we said they are in the sarcoplasm. So this is sarcoplasm. This is space is a sarcoplasm. All these are calcium. Once calcium bind in here, all right, what it does, it pulls, it changes the conformation of the tropomyosin, and it moves it on the side. So I'm just going to move it on the side now, so you'll see it a little bit better. Now it's moved on the side. See? Now I'm going to erase the other one. There. So now you could see clearly myosin head can actually attach to it because that's a binding site. So myosin head is now, boom, can attach, and then it could do the power stroke. All right, it could move and tilt the neck so that way it can go. Now, if you remove this calcium back up, same thing is going to happen. The tropomyosin is going to cover the binding site. This head is going to go back down, and basically tropomyosin is going to go back to the original formation. All right. So now that you understand this activity, we're going to put it in a step-by-step and take a look at exactly what happens. By the way, this green-like structure is called troponin. So calcium can bind to troponin, and as a result of that, tropomyosin can move. All right. So let's go ahead and take a look at a step-by-step formation and see what happens. So I think I have that on my notes actually, so we could just go ahead and take a look at it. So cross-bridge cycle, 1, that's exposure of actin binding site. In other words, calcium coming from the SR binds to troponin, and it moves the tropomyosin. So you could see calcium right here, right, coming in, binding to the troponin. This is troponin, right, and it moved the tropomyosin. That's step number 1. So at this point in time, binding sites for the actin are exposed. That's step number 1. Step number 2, cross bridge formation. Now, myosin head, you could see it in red, are rising up and attaching to the actin because they are available. Binding site is available, they are attached. That's number 2. Third step, power stroke. This is when the adenosine diphosphate departs, and that transfers of energy, which is a potential energy, to a mechanical energy. In other words, the head of the myosin tilts or bends, and it causes the actin to move. And also the sarcomere at this point in time are shortening. Basically, what is happening is the myosin head that is already attached, they are tilting. So here's the myosin head, now is tilted. All right. Of course, they are attached to the actin. Now, it's not just one, other myosin head can do the same thing nearby as well. Okay. So if we go ahead, this is called a power stroke. If we go ahead and take a look at this diagram right here, right, you could see many of the thick filaments, all right, they start attaching here, all right, they start attaching here, right? They start attaching here, right? Same thing here. They start attaching here. So once they got connected, all right, they can tilt the head. Once they tilt the head, you can see what happens. You know, this area right over here is getting shorter now because this area right over here is just start vanishing. And yet, if we go ahead and take a look at, let me see here. You see, over here, there is a little bit of space. Right here is a little bit of a space. So these actin filaments, they're going to come towards the center. And this red over here is going to go more in here. So the whole system, so pretend the Z disc is now here. See? All right? And pretend this Z disc is now here. Now take a look at the sarcomere. The sarcomere would be from here to here because it's from one Z disc to another Z disc, which is shorter than here. And when you're looking at many, many sarcomeres together, they're all getting shorter. Muscles is getting shorter, folks. All right, take a look. Each of these small units or sarcomere, you could see here is one sarcomere, here's another sarcomere, here's another sarcomere. So when they're all getting shorter microscopically, this muscles is getting shorter. And that's what muscle contraction is. All right. And we are going over the steps of cross-bridge cycle. All right, so what we were at the power stroke. Next up, we got myosin is attached to the actin again. We need to disconnect it. In order for the disconnection to take place, you need energy, ATP. That comes from the mitochondria again. So on the myosin head, if you recall, all right, not only you have one area for the binding site, but you have another area for the ATP. That's right in the head part right here. So if you have the ATP, as soon as the ATP binds in here, it changes the conformation and they get disconnected from the actin. All right. So that's step number 4. Step number 5. All right. So here is disconnected. ATP binded in the now is disconnected. You could see ATP binded, it got disconnected. Step number 5 is called hydrolysis of ATP. Hydrolysis, if you recall, we mentioned lyse with water, break with water. In other words, the water comes in and breaks the head, or I should say, hydrolysis of ATP breaks those phosphate bonds. Water comes in and break those high energy bonds. As a result of that, energy releases. And now you're going from ATP to ADP. Adenosine triphosphate goes to adenosine diphosphate. Adenosine triphosphate, we mentioned you're on top of the hill. All right, adenosine diphosphate, you're coming back down. So there was a potential energy in the phosphate bonds that once it's broken, you get the energy. This energy is enough to bring back down the head. Because remember, it was connected, it got disconnected. Now you got to bring it back to the high energy position so that that it could bind again. All right, that's the idea behind it. If this is too advanced for you, or you haven't looked at any of the lecture that I gave you before, give yourself some time, review the notes, look at many of the videos, read the material, read the chapters, do whatever you can on your part, and pretty soon is going to start making sense. If you've done that already and things are making sense and you understand, but you have questions here and there, you're in a good hand. In other words, now pretty soon you could ask your question, or everything is kind of like a review for you. All right. Step number 6. Remember, calcium was got out from the SR. So we need to put it back. Calcium is removed from the troponin, and they need to go back to the SR. All right, so if you recall, here was the SR. It had a lot of calcium inside, for example. All right, and during the action potential arriving, the calcium got out, so some calciums are in here now. All right, we need to put this calcium back in there, so somehow we need to take them back inside. All right, and as calcium comes off of the troponin, again, tropomyosin goes back to the normal position. This is important because you wanted to perform the muscular contraction again. All right. So, how do we do this? How do we place the calcium inside? Remember, calcium is charged, and those are more voltage gated channels that opened and this calcium got out. All right. But now what do we do? Is there any voltage gated channel on this side from the sarcoplasm to go inside? No. So calcium literally cannot go inside. Why not? Because they're charged. So under normal condition, we cannot get the calcium inside. This organelle has its own membrane. It's a membrane-bound organelle. And the calcium cannot go inside. However, there are a special pump in there. These pumps, all right, they can pump these calcium, 2 at a time actually. All right, so it's a good example of Symport. All right, 2 at a time, they can be pumped inside. But which side do we have more calcium, in the sarcoplasm or inside of the SR? Always it's inside of the SR, you have higher. So you might say, wait a minute, I thought some calcium went out. Yes, some calcium went out, but not the whole thing. You have a lot more calcium inside as opposed to the sarcoplasm. So the concentration is low in here, but it's high in here. So it's not favorable to go from low to high. So right away, you could see that this is not favorable, so they don't even want it to go inside. But you need to fill it up again, all right? Why? So that upon another action potential, calcium can be released. They can, bind to the troponin, move the tropomyosin, and again, cross-bridge cycle that we are discussing right now. But this calcium needs to be removed. And there are a special pump that they activate using, again, ATP. ATP is needed. Once you break down the phosphate bonds, energy is released, and that's enough to take the calcium from low to high. So I would say this phenomena right here is active process right here because it requires ATP. All right. So right away, when the calcium is departing and being removed, tropomyosin is going to cover the binding site again. And myosin, no longer can bind to it. All right. So here you can see the calcium is being pumped into the SR. I want you guys to think about inside of the SR, which you have a lot of calcium, think of it as a bank. Inside of the bank, you have a lot of money, all right? You may withdraw some money. That happens when you put in your code in here. That's what happens when, action potential comes in and voltage gated channels are opening, but they're not going to last because action potential passed. So some of the calcium is getting out, that causing the contraction, but now you still have more money in the bank. Even with you withdraw, bank always have more money. You go to work, a lot of you guys not working right now, but let's say you are, and then you need to deposit it into your bank. And this is kind of like a deposit in your bank, all right? Money in the bank, all right? So that would be pretty much your last step process, and we can kind of like take a look at this phenomenon also using animation. Let's discard this and take a look at the animation. And this is called uh, cross bridge formation. Cross bridge formation. Let's take a look at that.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere.
You can see many sarcomere. So look, these are the Z disc right here and these are right here. And notice when you see the sarcomere is getting smaller, look at the other ones. They're all getting smaller. So whole muscle getting smaller. See, now it's relaxed.
A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules in thin myofilaments.
Notice that the Z discs are here. Notice this is space, which is also known as the edge zone you learned in the anatomy, is vanishing. All right. And you could see how the sarcomere is getting shortened. So the muscle is contracting right now. These little dots are actually calcium. And notice, like, you know, the little heads are attaching from the myosin, which is red, to the actin. If you can't see it, it's going to get expanded right now. So in this video, they're talking about only 4 steps. Remember, I have 6 steps for you guys to remember. They did not count, like, you know, the calcium coming in from the SR as 1 step, and they did not count the calcium putting it back into the SR as another step. So for those reasons, this video is only talking about 4 steps. Your book can actually talk about 6 steps. Doesn't matter, just know the sequence of event, so that way you know what's going on.
The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin.
All right, so they're coming in from the SR. You could see some of them in here. This is the tropomyosin, and this is the troponin. All right, in my figure, I showed to you, like, you know, in a green color, if you recall. So they are actin filament because they are thinner. Now, these are the G-actin or globular protein known as actin. If they are made out of 3 protein molecule, why is it the whole thing called actin thin filament? Because it's thinner, that's all. And the majority of these are these actin. All right. So on myosin, there are a lot of these head that is bulkier, look like a golf club, and these are the handles, and there are many of them. So you'll see that in here as well.
This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin.
Notice that that was the first step. In other words, the calcium binded to troponin, and now you can see clearly how the tropomyosin is pushed on the side. Now take a look at this dark part. These are the binding site, okay? So the head part can actually bind in here. Let's take a look.
Allowing the myosin head to bind actin and form a cross bridge.
Okay, so in our sense, that was step number 2, kind of, right? So adenosine diphosphate departs, the bond get tighter, and the powder stroke is happening, all right? So pretty soon, this head is going to be tilted. Then ATP comes in and binds to the head and disconnect. Then hydrolysis of ATP, bring it back to the high energy state. And then last one was the transport this calcium back into the SR. So let's go ahead and continue to see what is happening.
Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps.
For our sense, it's going to be 6. So I'm just going to revise this step. Step number 1, calcium releases from the SR binding to the troponin, move the tropomyosin. Basically, this active site become revealed. This is a step number 2.
Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate.
Now you could see how the myosin and actin, they are crossed with one another, forming a bridge.
Is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4.
So notice that they're no longer connected. ATP binded to the head, they come disconnected, and now you have to kind of like do the hydrolysis or reactivating the head position so that they could bind to another one.
Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other, and the sarcomere shortens.
You could think about this as you lifting your dumbbell. All right, notice like, they are attaching in here, and the sarcomere from here to here is getting shorter. Don't forget, the last step was to pump the calcium actively back into the SR. That also requires ATP.
This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
All right. So there are a lot of other videos you guys can watch on YouTube and take a look. All right. Here.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
All right. So we went over the steps of cross-bridge cycle. We could go over part 2 now. All right, so this is a kind of like a summary of what just happened. You have the axon that is bringing the electrical impulses or action potential. In the synaptic cleft, there are acetylcholine that are released, binding to the receptor, acetylcholine receptor. There are ligand or chemical gated channel. They open up, ions, they go back and forth, and that causes the action potential to, or voltage changes to occur inside of the muscle. This action potential, they will propagate. What that means is they're going, all right, away from here, and they're going right into the T-tube, which is the pathway for these electrical impulses to travel and reach the SR. You can see the SR right over here containing a lot of calcium. There are voltage gated channels in here, as you can see, all right? And as soon as the voltage changes, this pretty much becomes open and the calcium is released. All right, now calcium binds to troponin, move the tropomyosin, and pretty soon you could see cross bridge formation is occurring. All right, then there is a power stroke, then there is a ATP connecting. So basically, they are disconnecting the myosin head and the actin. They are disconnecting, and then after that was the hydrolysis of ATP, bring back the head to the high position area. And then what happened is the obviously removal of calcium back into the SR. So eventually this calcium, they need to go back. That happens from ATP. Before I go on, I wanted you guys to think about mitochondria inside of the muscle, all right, and ATP in general. The whole events requires a lot of ATP. Think about it. First of all, the nerve exocytosis. Look at the mitochondria. That requires ATP. Also, the sodium-potassium pump. There are a lot of sodium-potassium pump. They pump 3 sodium out and bring in 2 potassium in. All right. There are all over the places 3 sodium out, 2 potassium in. So remember that this pump is constantly pumping. All right. This was a primary active transport, if you recall. So this is important because the concentration of the sodium will be high outside and concentration of potassium is going to be outside. But vice versa, concentration of sodium is going to be low inside and the concentration of potassium is going to be low outside. So when the channel protein starts opening, such as this calcium voltage gated channel, they go to the opposite direction. So in other words, a lot of sodium is going to go from outside into the cell. Is going to cause the cell to depolarize, in other words, get more positive. And when a lot of this is exiting, like potassium is exiting, then what happens is you have higher amount of potassium inside, but if they're exiting, they will have lower one. So again, you're taking out some positive ion outside of the cell. So these ions going back and forth, it causes voltage changes, and these voltage changes is important for controlling mechanism of dealing with the contraction, what you need to do, etc. All right, so let's go ahead and take a look at what happens next. All right, so this is the second part, and the previous parts of the second part we already discussed, so we're jumping into it. If you're trying to sort out which PowerPoint it is and so on, this is the part B, 9.2, and we are on the slide number 32. Because we already covered, well, actually, no, this is not accurate. We're not on number 32 because I added a lot of blank slide there. So no, that's not accurate. But it's right after this picture right here, okay? All right, so now there are 2 kinds of contraction. One is an isometric contraction and the other one is an isotonic contraction. Isometric contraction increases the muscle tension, but muscle does not shorten during the contraction. That does not mean the muscle is not working. Say, for example, you're pushing on the wall. Your biceps muscle is not going to get shortened, all right? say you're holding your book, maybe perpendicular to your body, all right? How long are you going to be able to hold that book? There are certain muscles that are involved in that. So those are the isometric contraction. There are also isometric exercises there too, all right? And then there is a isotonic contraction. Isotonic contraction decreases the muscle length. So this is kind of like now you're lifting up a dumbbell and your muscle is contracting, all right? So we call this like, during the flexion, for example. Think about when your arm flexes, all right, your biceps muscle is contracting, right? It's getting smaller. So whenever the muscle getting shorter, all right, that's called the isotonic contraction. All right, so let's go ahead and take a look at a motor unit, see what it is. All right, motor unit is a motor neuron that I just explained to you coming in from the brain, and it's also connecting or communicating with a particular muscle. All right, so they might be communicating with several hundred muscles or few of the muscles. It really depends. Now, whenever you are controlling fine movement, such as moving your eyes or moving your fingers, etc., then you have a smaller motor unit. What does that mean? Is that a bad thing? No, it's not a bad thing. You have a smaller motor unit because precision is what you're looking after. If you're driving, you want it to look around you to see where is the red light is going to turn in green, who is passing, for example, a pedestrian crossing the street. You are kind of like monitoring a lot of different things. For that, you require precise movement. There's got to be less muscle. There's got to be less motor units because, again, nerve controls those muscles. And you actually name them if you remember them. Oculomotor, for example. There were like a lot of rectus muscle, lateral rectus, medial rectus. So a lot of those muscles are important in the eye movement, basically. But also fingers are like that. They have a smaller motor unit. Then when you have larger motor unit, and what is the advantage of it? The advantage of it is that you have a lot of power behind it, all right? And probably the strongest muscle in your body are this type, all right? However, their motor units are pretty large as well. I mean, there are several of them, okay? And, so during contraction, you could see what is happening. Here's motor unit 1. It looks like maybe it's in, I don't know which color it is. But let's say motor unit 1 happened to be the black one. So this one. And motor unit number 2, that's number 2, that's number 2, that's number 2 as well. So these are all number 2. What does it mean by number 2? It just means it has more muscle fibers. You could see that now, right? These nerves controlling more muscles. Versus motor unit number 1, you notice that it's the black one in this case, or I don't know, darker blue. And you can see this one is that, and you can see this one is that. So if you ever go to the fair and they give you like this hammer that you have to hit this platform, and there is a bell or maybe an iron rod, it goes upwards. And if you hit this platform really hard with your hammer, you can go all the way on top and hit this right here and win a stuffed animal, like a Pikachu or something, right? So the concept is this: when you put in a lot of muscles behind and stimulating at the same time, then what happened to your muscle? Your muscle is going to grow bigger, stronger. All right. So you're doing it every day. You're even stronger than some other individuals and so on. Right. So that is really important to understand the idea of the motor unit. All right. So, uh, of course, uh, bulky muscles such as your quadriceps muscles and so on, they have a lot of power behind it, but you don't get as precise control like the eyes and the fingers, for example. Uh, but because there are a lot of different motor units, it allows you to recruit them together at times, and you could get even more contraction. So going back to the fair, imagine that you hit the platform with the hammer or a huge rubber mallet. What happened is the weight might go a little bit up, but this is as if though you only used motor unit 1. All right. If you use motor unit 2, because motor unit 2, you could see there are more branches right here. The one is on, here's 1, here's 2, here's 3. Okay. There could be probably a lot more. So now you're getting more nerves is going to and stimulate more muscles at the same time. You get it up to here. But if you wanted to hit the top and win the prize, then you have to stimulate both of them at the same time. Motor unit 1 and motor unit 2 has to work together, and all of them is going to contract, and obviously that will give you the highest power. All right. So this is, let's go to the particular experiment so you can see what's going on. All right. So if we go ahead and remove a muscle from a frog leg, this could be a gastrocnemius muscle. Let's say you get the frog and you kill the frog. Right away, you take out a muscle. You tie up one end of the tendon to a stationary position. You tie up the other point of the tendon to perhaps a device known as transducer. All right. So this transducer collect the data and put your results on a TV. All right. Now, if we zap this with the electrode, which is hooked to the voltage generator, which voltage generator is hooked to the, electrical outlet. So when you're zapping it, this travels inside of the muscle. As it's zapped in here, the muscle contracts. That is if there is enough voltage. If you put in low voltage, you may not see anything. If you see high voltage, you might see something like, boom. If you make it even higher, you might see it like boom. You make it higher, it might see boom. All right. So every time you zapping this muscle, you're getting a different results. The idea is when you are zapping it with the higher voltage, then more of these voltages is going to go to the more of those voltage gated channels and such. So basically what happened is more muscle fibers get recruited. As a result of that, you have a stronger contraction. All right. This is called a muscle twitch. All right. So each muscle twitch consists of 3 phases. Consists of 3 phases. Right now we are concentrating on muscular twitch. Here is a good example of the muscular twitch. The very first part, all right, is called a latent period. The second part is called period of contraction. And the third part is period of relaxation. Interestingly enough, all right, when you're looking at the, I wanted you guys to picture what you just learned in the cross-bridge cycle and relate it to each of these cycles. Then your understanding and the depth of understanding is going to be tremendously impact your learning ability. All right, and understand what's going on better, in my opinion. So notice that when we zapped, in other words, here's the muscle, we zapping it, and because this muscle is hooked to the transducer and hooked to the computer monitor, on the computer monitor we see the twitch, muscular twitch, right? But when we zapped, that's at the 0 time. So x-axis is time, y-axis is tension. Tension means force. If the force or tension is higher, then there will be a greater contraction, or vice versa. All right, so if we are zapping it at the 0 time, notice that the highlighted area, let's see if I can expand it. Notice this highlighted area right here is called latent period. All right, I wanted you to think about what do you think is happening, because there's no contraction. In other words, going back to the muscle fibers and myofibrils, there's no contraction there at all yet, up to this point. At this point, yes, there is a contraction, more and more and more contraction up to here. And then after that, it starts relaxing, okay? Because again, we discussed that muscles have excitability, muscles have extensibility and contractility. All of that makes sense. So let me go ahead and tell you guys what is happening in here. Latent phase, also known as a lag phase, lag phase. All right. All the steps before power stroke, remember the power stroke? Basically happens in the latent period. In other words, calcium is releasing, calcium has to bind to troponin, move the tropomyosin, myosin head has to bind. Up to this point, still we are in latent period. It happens quick. Electrical impulses goes really fast, all right? Calcium release, fast. Calcium binds to troponin, fast, all right? Move the tropomyosin, attachment of the myosin head. Up to this point, you got latent period. There's no contraction, there's no movement. Here were the actin filament, and here you have the myosin head is attaching to them. They're not moving, yet we're slowing down the time. All right, after that, what is happening in the muscular contraction? Well, now there is a power stroke. During power stroke, that's what happens, right? By the way, those ding song are my emails. Keep sending. I thought I turned off the notification so it doesn't bother us. Maybe I forgot to do it right now. Anyways, so muscle is getting shortened during the period of contraction. Think about a spring. Here's the spring inside of your pen. If you push towards the side, all right, they get smaller. If you push harder, they get smaller. Then you push harder, they get smaller. But you get to a point that no longer can get smaller, right? So this is kind of like really now compressed. But after a little while, you know, you cannot push it from both sides anymore. They're not going to get smaller because they reach their maximum. Same thing with the muscle. When the muscle is contracting, you can only contract it to say certain, you know, So if I zap this muscle, for example, with, let's say, 5 millivolts, all right, you might see something like that. If I zap it with 10 millivolts, you might see something like that. If I zap it with 15 millivolts, all right, so the twitch is getting higher and higher and higher. But if I zap it, let's say 20 millivolts, let's say I'm getting this. If I zap it at 100 millivolts, I still getting this. What does that tell you? That means maximum has reached. Cannot make it more contracted than the max. All right. I should have put the arrow in here so it tells and emphasize that it's on the contraction phase. All right. So after that is the relaxation phase. And the relaxation phase, as you can see over here, the tension is reducing. And erase this. Right, the tension is reducing as the time passing by. All right. So what are the events that during the, because the period of contraction is simple. These are when all the myosin head starts binding, and pretty much they are keep contracting or tilting their head doing the power stroke, and the sarcomere is getting shortened. What about the period of relaxation? What if after that, what do you think is going to happen? Let me go ahead and pause the video, or maybe I should just ask you, what do you think is going to happen during the period of relaxation in terms of the cross bridge cycle? Remember, after the power stroke, what happens? Anybody wanted to take a guess?
When the calcium goes back?
No, not yet. Yes, that happens too, but not yet. What happens right after the power stroke?
Hydrolysis of
Dealing with
ATP.
Okay, ATP binds to the head. That's correct. So what happens is they get disconnected. All right? All right? So once they get disconnected, there is no more contraction now. Now, all of the events after that is during the period of relaxation. So some of you guys said that already. So for example, ATP hydrolysis. Yes, that happens in the period of relaxation. After that, re-energizing the head. All right, that's what happens. After that, calcium departs from the troponin, moving back into the SR. Yes, all of that is happening during the relaxation. All right, so that is what I wanted to talk about, and that would be the muscular twitch. Let me go ahead and give you guys a little bit time to recover and resume recording. Okay. So we were talking about the muscular twitch and the difference between muscular twitch and the wave summation. Wave summation is just what happened is, you are, let me go ahead and, thank you for reminding me, Taylor. Appreciate that. All right, so in the wave summation, because the calcium already there in the sarcoplasm, and it takes time for the calcium to keep pumping back into the SR and be removed. So when the second one comes in, this is the second one, all right, more and more calcium is going to be available. So therefore, more degree of contraction. That's why this one is higher than this one. Right? And same thing goes with the next one. All right. So that's the difference between them. Now, if you zap the muscle at the closer interval, you're going to get the same thing. So this is the same thing. The only thing is different is you notice they are closer at one another. So you're still getting more and more. So the only difference is the zapping timing is at higher frequency. It's closer to one another. So this is called incomplete tetanus or unfused tetanus. What if we zap them so close to one another, they don't have any even partial relaxation? So in other words, muscle contract. So at the beginning, this is the muscle. And then once you keep zapping it so fast, boom, 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 the zapping, it keeps coming in through the electrode, and you're zapping these muscles like every 2 milliseconds apart from one another. then there is no time for relaxation whatsoever. So you could see there is no relaxation. There is a lag phase right here, and there is a period of contraction, which is from here all the way to here. You might say, well, isn't that relaxation? Well, it is, but remember the zapping is stopped at this point in time. There's no more zapping here, right? That's why they are relaxing. But during the zap time, you didn't see anything going down, right? That would be the relaxation, but it's not. So the muscle is not relaxing, right? And this is called complete tetanus. Now, interestingly enough, we go ahead and take a look at, zapping. I'm not going to draw the arrow. I just put 3 dots and just like same thing all the way here. In between, you could fill it up yourself. So it went up. Here's the lag phase. Now they reacted. Why didn't they react it in a 0 millisecond, let's say? This is millisecond. How come we didn't put it right over here? All right. Again, remember, it takes time until the electrical impulses release the calcium. Calcium bonds to troponin, move the tropomyosin, right? It takes time, all right? So the period of contraction is actually starting when it goes upwards, right around here, right? That's when it's keep going up. This is the time that the power stroke is starting, actually, right? Right, so it's going up, and it's going up, and then it plateaus, and then it's coming down. And if you continue doing that, it keeps coming down. But wait a minute, you keep zapping it. How come it's coming down? They're not responding anymore. In fact, if you continue, maybe I should make it lower right here. It's gonna go all the way to the end. Muscle is not responding, but we're zapping it. Why not? Here's the deal. If you keep on zapping the muscle keep on contracting, right? And there's a lot of ATP that are diminishing. ATP is the source of sodium potassium pump we discussed. ATP is the source for pumping the calcium back into the SR. ATP is the source for, disconnecting, all right, hydrolysis, putting the head back into the energy, power stroke. So we use the ATP a lot. If we're running out of ATP, the muscle is not responding. Earlier, I told you when you are running, at the beginning, you might have a very high speed, but as you keep on running, you're running out of ATP. You're sending a signal to your muscle, telling it to go and run as fast as you can, but they can't respond because your ATP is diminishing. All right, so what do you need to do? The only thing you can do at this point in time is wait until ATP replenish. All right, once the ATP replenish, then you're okay. All right, let me pause for a moment. I wanted to get to another YouTube video for you guys. On the survey, some students say, I really like the way that using the YouTube video. So here and there, I use it. So let's go ahead and take a look. Bear with me. All right, the quality is not that great, but that's okay. Let's just go ahead and take a look and see what happens. It's a short video. It's a marathon, and they are running and they're getting tired.
The finish line is so close, and someone is so close behind. After more than 140 miles, it's come down to less than 100 feet. to that damn line. Just yards behind, her stalker appears. It's Wendy Ingram.
Okay, as you saw in the movie clip, uh, they ran out of energy. The brain sending a signal, the signal that we discussed in neuromuscular junction, all right, going from the motor neuron, from the brain, going into the muscle. They're releasing their acetylcholine. Acetylcholine is binding to the receptor. The receptors are opening, those are the ligand gated channel. Ions are going back and forth, right? Transmitting their neural impulses into the muscles. They're reaching the calcium voltage gated channel on the SR. Calcium is releasing. Calcium is binding to troponin and tropomyosin moves. All those activities taking place. But when you don't have energy, none of those is going to be working, because at every level that ATP is involved is a shortage, because you're using it for a lot of them for contraction. Where are you going to get the money if you withdraw all your money from your bank account? You're not going to have any more left if you keep withdrawing. This is kind of like same understanding. However, if you withdraw some money, you spend, and then you get some money somehow, and then you are depositing it, you still have some to operate, right? So this is kind of like a similar concept I want you guys to think about. All right. So sodium, potassium pump, there's no energy. They can't restore the balance. You got ATP, you don't have anymore. How are you going to disconnect? The muscle is not working. How are you going to pump the calcium back into the SR? So that yeah.
Are we still on the video, or are we on slides or something?
You don't see anything right now?
I still see the video. I don't know anybody else.
Yeah, I just paused the video, so I was just explaining that.
Oh, okay. I'm sorry.
No, it's okay. Thank you for letting me know. If you want to go to slide, we could go to slide. But I was just like talking about what happens in here. The muscle is not responding. That's what is happening. Even though you are zapping the muscle with the same thing in the laboratory, you notice like the muscle is not responding because you need ATP, and they're getting exhausted as far as the ATP goes. Same thing in the video, right? So this is called fatigue. Muscular fatigue happens when the muscles are no longer responsive, all right? And there is also something known as a psychological fatigue, that you think you're tired, but you're actually not. You could keep on going. So, but physiological fatigue, when your ATP no longer there, or you have much reduced level and the muscle is not performing, all right?
Question, sir.
Yeah, go ahead.
Is the lack of ATP the same thing as when you have a lot of lactic acid build up in the muscle?
It can. The lactic acid, I'm gonna talk a little bit about it, but yes, the balance, homeostasis gets disrupted. There is a lot of lactic acid starts accumulating. It can lead to the acidosis, blood acidosis, and other problems. Yes, absolutely. Okay, because the system is shifting from aerobic to anaerobic, and I'm gonna talk a little bit about that. So I might as well just talk about it now. So there are, in terms of metabolic energy and so on, aerobic mechanism is the most efficient way to get the energy or the ATP. In other words, this is a little bit biochemistry. Those of you that took biochemistry or maybe you have a little bit knowledge from before, that's great. If not, I'm not going to take time to go over in detail, but I will paint the picture for you. And whenever you take this course, you go over the detail. See, we get our energy, majority of it from glucose, all right? But the glucose, you can't just get the energy by just you have to break it down. And there is once you break it down, then it releases energy that you need. But in order to break it down to get the usable energy that is ATP, right, they have to go through a process known as glycolysis. Glucose breaks, glycolysis, it breaks. And then it changes to pyruvic acid. Again, once the pyruvic acid is reached, then this can go into the mitochondria along with other macromolecules. And when the oxygen is present, all right, you give off carbon dioxide. We discussed this already, site of cellular respiration. You're making water, but also you're making a lot of ATP. And we mentioned this. This is like 36 ATP per one glucose molecule. A candy bar has millions of these glucose molecules. So you think about how much ATP you could make. But this needs to have oxygen present, right? And that's why we call it the aerobic mechanism, right? The duration of the energy is hours. You could go on for a long time. However, sometimes athletes push themselves to the limit, for example, like the video that we discussed. And as a result of that, you are taking so much oxygen and carbon dioxide is building up, but your respiratory rate work and operate, even though you're working fast and so on, it still is not enough oxygen for this maximum ATP that you need and require to make. So the system changes to anaerobic mechanism. Anaerobic mechanism, you can produce a lot of lactic acid. Again, I'm not going to go over the mechanism of it, but it can acidify the blood. That's not a good thing. But you can get some ATP from it, all right? The ATP that you could get, it might last you about a minute or so, which is not much, but maybe enough to, carry you out. And the last one that we discussed that you get very little ATP is by, breaking the keratin phosphate, which is CP. And as a result of that, you could put in one of these phosphate to the ADP, and you're going to get one ATP. So again, look at it. Keratin phosphate, 1 ATP. Anaerobic, 2 ATP. We're talking about, per reaction, in this case, per glucose molecule, and then 36 ATP. So you can see that this is the most efficient way in order to generate ATP. All right. Now, let's go back and talk about trippy. Trippy, or a staircase event, don't make a mistake and think that it's a wave summation. In a sense, the wave summation, you get the second contraction is more than first, and so on. The third more than second, the fourth more than third. So this is kind of like a stairs that you're going up the stairs. See, it's like a stairs. However, in wave summation, you got the partial relaxation and then it went back up. This one is a complete relaxation. So the frequencies are not as apart as you think. all right? They're far enough so that you can contract and then zap, and then you're going to go next, and then again complete relaxation, and then again zap, you get a more contraction. The these are all zapped at the same voltage, say for example 10 volts. Then how come you're getting a higher contraction compared to the first one? They're both 10 volts. Yes, voltage can increase the current, but we didn't change the we didn't change the voltage in this case. Then why is it that we're getting more and more and more? Again, the reason is two reasons. One, calcium availability. Once you contract, calcium releases in the sarcoplasm. They have more chances to bind to the troponin and move the tropomyosin. The second one comes in, again, more calcium. It takes time for the calcium to pump in, all right, back into the SR. And also when you are contracting the muscle, it generates heat, and apparently the enzyme that requires to do this during the muscular contraction and so on, all right, they work more efficiently when there is a temperature slightly is above that. And contraction produces kind of like when you rub your hands against one another, generating heat. And some enzymes, they have a specific heat, a specific temperature that they work best. And apparently those enzymes work best under a slightly higher temperature. So it's always a good idea to warm up before you exercise, all right? When you warm up, you're doing a task completely. Doesn't matter whether it is a stretch or jumping up and down or jogging and so on. When you're looking at athletes, let's say basketball players, soccer players, tennis players, you name it. Before the actual event, they warm up. They jog, they kick the ball, they shoot, just very relaxed way a lot of time. Why not just go full force then? Why not? Why is it advantageable to just do it a little bit mild, so when they get to the event, they actually will have, the muscle will perform the best because they could get maximum contraction. As you can see, in this case, that's the max. You cannot keep going up. Why not? What do you think? Anybody want to guess or try to answer it? The question was, why not just, instead of warming up lightly, why not like run like crazy to get really warmed up and then go?
Maybe they will fatigue quickly.
Exactly. So if you do that, good job. So if they do that, they're going to use up a lot of ATP. And then when the event comes in, they're going to run out of ATP. Right? The muscle might perform at the maximum level, right? But they're going to be running out of ATP. Right? So you wanted to monitor it, so not to do too much and not to do too little. So that way you have, enough calcium in the sarcoplasm. And hey, sports physiology is integrated in the sports now. Everything is supposed to be so precise for different bodies and so on. So it makes a big difference to be first person in swimming or finish line, or let's say, fourth place, which you're not going to get any award. All right. So for that, people are doing all kinds of things, integrating sports science as well to making sure that they always, perform at the peak level. All right. Okay, what we're finally going to end up with is two contractions that we discussed. One was an isotonic contraction, which you can see because the sarcomere is getting shorter and the muscle is getting shortened. In this case, it's a 30 kilogram weight that you could overcome. Muscle getting shorter. All right, tension is the same. Iso means the same. Tonic refer to the tension. All right, so during the isometric contraction, all right, iso, same, metric, measurement is the same. So tension is increasing, but the muscle, they do not get shortened or lengthened, but they're still working. So let's take a look. This is in case like, pushing on the wall or you're lifting some object that is impossible for you to overcome, let's say a big piano by yourself. All right, so you can't do that. So therefore, the muscle is still working, but they're not getting shortened. These are called isometric contraction, when the length stays the same. All right. So to finish up this lecture, we learned about the physiology, and again, there will be some parts that I will post up for you again. But we looked at the cross bridge cycle in this video. We looked at the twitch phenomena, the different phases of the twitch, and the wave summation versus incomplete and complete tetanus, followed by the fatigue. What are the sources of the ATP? Where do we use the ATP? And two kinds of contraction. Those were the summary of this particular lecture. We also talked about the motor unit too. All right. But in a nutshell, we can understand the physiology of muscles, skeletal muscles, and try to apply it, implement it in our daily life as much as we can. When you are at the relaxed stage, still your muscles is being stimulated. All right, these are called muscle tone. All right, so when you wanted to remove them or contract the muscle, they respond quickly. So there's little tiny electrical impulses that are being sent and making the muscle slightly contracted at all time. All right, so they can respond quicker. When you are in emergency situation, all right, you can recruit a lot of muscles. Going back to this part right here. For example, something emergency, the house is in fire and you need to get out, right? All these motor units, they're going to stimulate all at the same time, all right? And they're going to all contract, so you're going to get the maximum response. In fact, if somebody timed you during the possible fire in the building, you might break your own record how fast you're getting out. All right, that's how fast you're going to be, because again, you're going to recruit a lot of these muscles. A lot of times during emergency, these muscles get recruited all at the same time. Motor unit 1, 2, 3, 4, 5, all stimulating your muscle. If you ever watch the weightlifting on TV, you notice they try to recruit all their muscles before they lift up huge weights, sometimes a lot more than their body weight, all the way on top of their head. That's really amazing. All right. You hear a lot of the stories. Guys, we have a lot of stories that we can think about. For example, a person was underneath the car changing the oil, and the jack just comes down. The car comes down on the person, and nearby, maybe his brother or somebody has enough power to lift up the car. Again, at that type of situation, you're recruiting more of these muscles. When you recruit a lot of these muscles all at the same time, all right, obviously your strength is going to increase. All right, and that's the idea for it. Sometimes we may not be able to recruit all these muscles, and as a result of that, the strength may not be there. All right. Another thing that I can think of that is more beneficial that you guys can think about in terms of exercises. When you exercise, your heart rate goes up, your blood vessel dilate, and you bring in nutrients to everywhere, including the muscle. So the muscle can grow better, they can have more nutrients better. So your heart is getting exercised, your skeletal muscle, having a lot of the nutrients. And people that are athletics usually are healthier. They don't get sick as often. You know, many of the individuals that overcome the COVID-19 were NBA players, you know, New Jersey Nets. Even I heard like maybe one or two people in Lakers, and they overcome it. Uh, Rudy Gobert, you know, all of those individuals that were like athletes, their immune system was um, perhaps stronger because when you have good blood flow due to the exercise, your brain gets more flow. You can think better. Your uh, most of the system works better because of uh, more nutrients traveling towards the areas and so on. All right, so a skeletal muscle is involved, heart is involved, you know, and also smooth muscles involved because blood vessel dilate during the exercise to, you know, dissipate the heat and bring in more nutrients, and again that causes blood vasodilation, which inside of it is a smooth muscle. So we began by talking about the three types of muscle. We ended by three type of muscles as well, and how also things can be related and beneficial to us. All right. At this point in time, let me go ahead and stop the video.